Recording in progress.
Hello, good evening, everybody.
Good evening, sir.
That's, I think, Sahas, right?
Yes, sir.
Yeah, now I can recognize from the voice. If you speak, I can recognize from the voice that who is speaking. Yes. All right, so I kept these drawings from the last class on Monday morning. We haven't finished the transpiration, and today we're going to finish transpiration. Right, just to summarize a few things from the plant. Number one, the transport system of the plants consists of xylem and phloem. The xylem transport water and mineral upwards. Phloem transport nutrients such as sucrose and amino acid upward and downward. Mostly both of them are organized together to make the vascular bundle, and we have seen how they organize in the root, how they organize in the stem, and that's how they organize in the leaf. So they make a continuous tube from the root to the leaf, that's the end, and they can transport substance up and down based on what they transport. Then I also mentioned about this fish tank story. So that's something to understand, that if we have a kind of a water container like this, and if you want to empty, you just insert the tube, and initially you have to take some liquid out. You have to suck it out, and then it just can go out continuously, and it can empty it. That kind of a special activity or property of the water helping to go. So transpiration, you can see again revise the cuticles, upper epidermis, palisade mesophyll cell, spongy mesophyll cell, guard cell, and the stomata. So that's a xylem and phloem organization. And what I said, the transpiration, the definition of transpiration is evaporation of water from the cells, example palisade spongy mesophyll cell, and you can see here they just come out and now they evaporate, and then they leave from the stomata as water vapor by diffusion. The definition of diffusion is movement of molecule from high concentration to low concentration. So this area is water vapor low, this area water vapor is high, then the water can move by diffusion, and here water escape because water is liquid. So liquid can, liquid, solid, and the gas, right? So that's a melting, and that's a boiling, melting at boiling. right? And this way is a condensation and freezing. So if the solid is going to the gas directly, it's called sublimation. But anyway, if you look at this word boiling, so liquid become gas when we boil. So we have to use, I mean here the boiling mean we have to use temperature. So water boiling temperature is 100 Celsius degree. But here it's not boiling, it's evaporating, because water molecule can detach from each other with the normal temperature, and they can move out of the other water molecules, and they become a vapor. That's called evaporation, right? Evaporation, and then they can leave out, right? So that's a transpiration. Can anyone check whether we have defined and we have written it, the transpiration?
Sir, we didn't write anything for it.
The definition? Did we write it?
No, sir.
No?
We didn't write anything.
This
Only
drawing?
the diagram.
This diagram?
Yes, sir, we drew it.
Okay, right. Right, then can you write the definition of transpiration? Definition of transpiration. Okay, transpiration is the loss of water vapor transpiration is loss of water vapor transpiration is the loss of water vapor through the stomata. Through the stomata.
Excuse me, sir.
Yes?
Sir, the xylem and the phloem is in the stem of the leaf, right?
Could you please repeat the question? Xylem and phloem?
Is in the stem of the leaf, right, sir?
Stem of
They
the
come
leaf.
from the stem. Yes, sir.
Okay, wait, this is stem. Do you, are you asking, so let's say this is a leaf. Are you talking about this part?
Yes, sir.
Okay, so I think we don't learn that kind of a thing. So it's a so here is the stem, let's say. Right now, this one Okay, so you know, we don't learn about the structure of the leaf. So this part has a special name, but not even in the textbook you can't see. I think it's not there. It's called petiole, right? So what is this is the stem, and now stem and the leaf attached to each other with the petiole. That's called petiole, and this middle the middle line is called midrib, and the other branching areas like this is called vein, right? Now the xylem and phloem, they are in the stem, so they are now branching to this part, and they are going to the midrib, and then they are going to these areas. So the xylem and the phloem, they are coming from the root to the stem. From the stem, it is attached to the part called petiole. The petiole continues as a midrib. Midrib is branching to make the veins. That means xylem and phloem is going all the way down to the end. Now you see that's the end. So this is the end part of the xylem and phloem. Otherwise, it has to come from the stem, petiole, midrib, vein, and the branches of the vein. So then it's distributed all over the leaf. So if you want to see the structure of the xylem and phloem, what you can do, take a big leaf like let's say mango, or a little bit other, even you can take any leaf, and then place in a book, inside the book, within the pages. After a few weeks, everything is digested, and the xylem and phloem you can see as a kind of a small hairlines, like organization in the leaf, that one. Sometimes in the nature also we can see on the floor. You can then observe the complete structure of distribution of xylem and phloem. So yes, they are in the stem, petiole, midrib, and vein, everywhere. Okay, so right, so now we can see xylem, and the phloem, so here it's a xylem, phloem, and here also we have a xylem, phloem, and here is also xylem, phloem. Okay, let's see how the water enter to the xylem. Now water can enter to the xylem this way by osmosis. So water enter to the xylem by osmosis. Again, the definition of osmosis is high water potential to low water potential through a permeable membrane. The enter of the water molecule is called osmosis. And here water leave as water vapor. That's a transpiration, right? Now how water goes up? Now it is something like this. If you open a tap, okay, Abhi's, wait a second. hello. Hello. මං අර ගෲප් එකට දැම්ම මේ මුකුත් මැසේජ් එකක් ආවේ නැහැ මං හිතුවා අපි අර නිවාඩු හින්දා මේ දවස් ටිකේ එන්නේ නැද්ද කියලා. දැන් කමන්නේ දැන් ෆිසිකල්ද ඔන්ලයින්ද පුළුවන් මේ ක්ලාස් එක පටන් ගත්තා. අයින්න මං අරින්නම් මේ ගේට් එක අරින්නම් මං ඉෂුව එකක් නැහැ. හරි හරි. 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 Okay, can you draw this picture until I start the class? Can you copy this one? Okay, now I will explain how water molecules move from the soil as a water molecule to the air, I mean atmosphere, right? So now how this happen in the root? The water can enter by osmosis, that is high water potential to low water potential, because water is high in the soil and initially less in the root. Then the water can enter into the root. Then more and more water enter into the root. What will happen? Pressure increase. Then the root pressure increase. So this root pressure usually move upward. That means this direction water can go up. Then it's called root pressure. As water enter to the cells, then pressure build up and the water can slowly move this direction. By osmosis, water is pushed from this side and the root water is going up. Then it come to the stem. There is something called capillary action. So capillary action is like this. So if you want to observe water behavior, so you take a beaker and fill with water. Then you can insert a small glass tube like this. So when you insert the glass tube, you never observe water stay in this level. The water will go up, actually stay like this. That means water can go up. So water can go up because of the capillary action. Just water molecules try to move upward, like mostly we can see it like this, something like this. The curvature is like this because when water molecule attached to the surface is trying to go up, just like a Spider-Man, they try to move up. That's a very special quality of the water, property of the water, and that's called capillary action. So then they can go up from this capillary action, which is also driven by the loss of water vapor from here. That's something like what we have mentioned previously. When you open the tap, then the water continuously go out. Same way, the water vapor move out, this one pull it up, and then they can always go upward, because when more water loss, they try to replace the water and they come from here. That is called transpiration pull. Now you see, this is the three stages or three method which can be seen in root, stem and the leaf. So which make a kind of a continuous water column upward. Then the water is always moving upward, then water goes out. But one thing you can still understand, if we close the stomata, then whole process stop, because they can't escape here and there is not going up and the root pressure not enough to open the stomatas. So in the night, the stomata close, transpiration will not take place. It's only take place in the daytime, because water is required for photosynthesis during the daytime. Nighttime photosynthesis will not take place, so this transpiration will not happen. So can you also add these three the type of actions in root pressure, capillary action and the transpiration pull?
Sorry, I did not understand what you said about day and night.
Okay, it's very simple. During the daytime, we all know photosynthesis take place. Nighttime, there's no photosynthesis. Photosynthesis need water, carbon dioxide and water. So how water comes to the cells? Through transpiration. Then transpiration happen only in daytime in order to continue photosynthesis. night time, there is no photosynthesis, then they don't take oxygen, and water is also not required. Then they close the stomata. Mostly nighttime stomata are closed. The daytime they are fully open. Nighttime mostly they are closed. So why transpiration happen in the daytime? Because water is needed for photosynthesis. Why it is not happening in the night time? Photosynthesis not take place in the night.
Thank you, sir.
Okay, can you write number 1? Number 1. Water enters to the xylem in the root. Water enters to the xylem of the root by osmosis. By osmosis. Then root pressure increases. Then the root pressure increases. And water move upward. And water move upward. Number 2, when water molecule enter into the xylem of the stem, water drag upward. Water drag upward due to capillary action. Capillary action. Number 3, water vapor leave out of the stomata. Water vapor leave out of the stomata by transpiration. This makes force called transpiration pull. Force called transpiration pull. Force called transpiration pull. Which make continuous.
Could you repeat that?
Water moves out from the stomata by transpiration, the force called transpiration pull. Transpiration pull which make continuous, right? Which make continuous water movement upward. Okay. Okay, next, the last thing we're going to learn here, factors affecting transpiration. Factors affecting the transpiration. Number 1, temperature. So temperature increase, so when temperature increases, the movement of water molecule going upward increases, because when at high temperature, when the temperature increases, the rate of photosynthesis also increase, right? So now when I mention when temperature increases, the rate of photosynthesis increases. Why? How can we explain that? When the temperature increases, rate of photosynthesis increases. Yes, I can accept that. That's a very common theory. When temperature increases, the particles move faster, and you will learn this one, maybe already learned, sometimes called kinetic energy. That means particles also always vibrate, like water molecules, air molecules, like things. And even a solid, the particles can move a certain pattern that is limited. And temperature increases, the particle movement increases. But how can you explain increased particle movement, increased rate of photosynthesis?
When there is warmer air, like may temperature or like the photosynthesis also increases when there is warmer air.
That's nothing to do with air at the moment, right? Okay, you have to learn this temperature and the molecules, like things, many times in chemistry and physics and biology. But when it comes to chemistry, physics and biology, the molecule movements you're going to learn in a very different way, right? So you will learn very nice thing in the future. We can actually bring down completely a zero level the movement of molecules. It could be, let's say, in a gas molecule, let's say. So gas molecule, we can stop movement of gas molecule. So if there's a container, this container filled with gas, these molecules move. These molecules move. Then what is the force actually they move? Temperature, heat. then how we can stop it? Simple, cool it down. So we have to cool it down to minus 273 Celsius degree. That is called absolute zero. This is called absolute zero, and then stop. Now you understand, we have to bring to such a cold temperature if we want to make a completely zero movement of molecules, gas molecules. Now that phenomenon is going up, like when you increase temperature, gas or water molecules, they move. Now when it comes to biology, what is important to remember, as you have to learn differently different subjects. So what is important in biology, not like chemistry, so what actually increased the rate of reaction? In biological system, what actually increased the rate of reaction? Without that, we cannot have biological system. We are not existing. That is, all the living organism, these molecules actually increase the rate of reaction. Otherwise, the rate of reaction is completely slowed down and organism cannot live. These molecules are called enzyme, right? So now you understand enzyme. So if this is enzyme molecule, you know, this is active site of the enzyme. Now when I'm talking about the molecules, like whatever molecules, this is a molecule. So usually we say when temperature increases, this can move fast and bind here. So then you know, 1 second it take, let's say 20 Celsius degree, it take 1 second the substrate bind to the enzyme. So at 20 Celsius degree, the substrate bind to the enzyme and it take 1 second. Let's say if we, and then they make the product. Probably have for
Recording in progress.
right. At 20 Celsius degree, the substrate, that's a molecule, join with the enzyme and then make a product. It take 1 second. So if we increase the temperature to 30 Celsius degree, then it may take 0.5 second. Now you understand, when we increase the temperature, the rate of reaction increase. Why is that? Because molecules can move faster. So temperature increases, then you know, if we want to elaborate, you're going to say when temperature increases, kinetic energy increases. Kinetic energy increases, enzyme activity increases. This you have to write like in detail based on the question. If the question carries 3 marks, then you have to write temperature increases, kinetic energy increases. Kinetic energy increases, the molecule move faster and it can bind to the enzyme faster and more products form. So here the temperature increases, the rate of photosynthesis increase. Okay. The photosynthesis increase. So I just told how it increased because enzyme activity increase, rate of photosynthesis increase and transpiration increase. So now temperature increases, transpiration also increases. Okay, now one factor, that's the first factor. Factor 1, temperature. So you can write it down, the first factor. Then
Sir,
yes,
for the factor, should you only write temperature?
temperature increases, photosynthesis also increases, right? photosynthesis also increases and transpiration increases. You have to write this: temperature increases, photosynthesis increases, transpiration increases. Or same way, can you complete this one? Light intensity. Light intensity increases. So light intensity increases, the rate of photosynthesis increases, because light energy is an important factor for photosynthesis. Then transpiration increases. Third factor, wind speed. So wind speed increases. What actually happening? The vapor take it away. So the water vapor can take away from the leaf, and it make a gap. Then the water vapor can come quickly. So wind speed increases. The water vapor move out faster, then transpiration increases. So wind speed increases, the water vapor remove, and the transpiration increases because more water vapor can come from the leaf. The last one, humidity. Okay, so I should explain what is humidity, because some of them may not, they may have no idea about what is exactly the humidity. The humidity,
The amount of water vapor in the air.
yeah, that's right.
The amount
That's
of
right.
water vapor
That's
in
the
the air.
amount of water vapor in the atmosphere or air. So then, what will happen? The humidity high mean the amount of water vapor is high in the air. When the water vapor is high, the diffusion is not efficiently happening, because too much water vapor in the atmosphere reduce. This is the only thing we can say, reduce the transpiration. Increases transpiration, reduce. Transpiration reduced. Temperature, light intensity, wind, humidity. So these are the 4 factors which can increase or decrease transpiration. Okay. So any of you getting the rain now right now, or how about your weather? Because we have a student from Anuradhapura, Kurunegala and Ratnapura. So anyone from Kurunegala, Anuradhapura, or Ratnapura, or Gampaha, Nigambo, yes, Yashra is here.
I'm from Ratnapura, but right now there's not any rain.
Okay, let's hope we have a good weather. I'm not sure, but coming up days, December month is not that. Yashra is there. Anyway, yeah. Okay, so today I also have a video, but this video I have to play it soon because I actually forgotten to say something or add something in the class. This happened because mostly you get a term test and loaded with the exams. We also get stressed up and we want to finish syllabus, we want to finish the more tudes and more questions. Then we just forget what we have to say, certain things. And I think I remember that we're going to do a question as a homework. Question number 4, I think, homework. Yes, question number 4. So, I have something real story which is related to the science, like what we are learning, most of the things. Now, we learned about the prokaryotes. What are the prokaryotes? Prokaryotes are bacteria. And you have to learn 2 bacteria in our syllabus, and we have learned these 2 bacteria. One bacteria is called pathogen. Why we call pathogen? They can cause disease. I'm pretty sure someone can still remember the name of the bacteria which cause disease.
Pneumococcus.
Yeah, we learn pneumococcus bacteria is a bacteria which can cause pneumonia, and it's a quite dangerous conditions. These all happen mostly the pneumococcus, the bacterial disease kill I think millions of people in 1920, around 1990, or it was actually 1919 or 1920. So 2019 we had a COVID, like after 100 years, and that was also a bit of a weird. So we go back 100 years with the timeline. I'm pretty sure it was 1970, 1918, like this range, 1917, 1918. There was a disease called Spanish flu. The Spanish flu is caused by virus, but when people got the Spanish flu, after that they actually die because of pneumococcus. Then this opportunity take pneumococcus to also enter, and if I remember, millions of people died. Right, that's a pneumococcus. And when we go back to 1900, why many people die because of bacterial disease? For example, again we go back to 1300. 1300, that was the most dangerous disease came to the world. I think half of the population died. I'm not sure. Half of the population died because that time people don't know 13th century how many people died, but they literally say half of the people died. and that was caused by a bacteria. That bacteria is called Yersinia pestis. As all people know that as a black plague. People know this as a black plague. Now, when bacteria come, that time people died. Now, that much people will never die in bacterial disease because we have medicine to kill the bacteria. Those medicines are called antibiotic. So we have a lot of antibiotics, we can kill bacteria, but virus we don't have medicine because virus do not have a proper structure. So this disease, okay, before we come quickly, we can mark, right? Part of digestive system, question number 4 of the tube. Question number 4, page number, don't have page number because these are the PDF file. I cannot enter page number. I can enter, but I had to do manually sometimes the PDF file page numbers. I don't have a proper program. So it's a question number 4 of this tute. So when you go to the last part, you have the questions. After the note, you have questions. Then questions start with number 1, number 2, number 3, number 4, and number 4 now. The part of digestive system A. It's a mouth. A is mouth, only starch digest. Starch digest because of the amylase enzyme digests starch into maltose. Fat and protein, no, you had to use the cross for fat and protein. Part number B is a stomach. Stomach, starch no, fat no, protein yes. That is the enzyme pepsin. Pepsin digests protein into a peptide. Then number C is the small intestine. So small intestine include the duodenum and the ileum. So they are all three digest starch, fat and protein, all digest. Part number D is the rectum. Oh, yeah, we can call it as rectum. There is no digestion. The cross for all three. Okay.
Isn't that the ileum?
Sorry?
Sir, in the ileum, none of these get digested, right?
Ileum, no, ileum I think more absorption. So mostly duodenum and the middle of the intestine, we can still observe digestion. It's like I would say like a duodenum. The first part is more, most of the digestion take place in duodenum. But when it comes to middle part, we can also absorption and digestion both. And the large intestine together, we can consider as digestion and absorption both take place. But the digestion decrease with the length increase, but absorption increase. But when they pointed out to C, we can consider as a small intestine. So that's a place all three digest.
Okay, sir.
I think you can draw this one by looking at the note. Draw a label diagram of a villus. I will not go to that to save the time. And you can check from the note. This is also some example to get an idea. In exam, sometimes you have to draw and label structures, but that is not common. Once in a while, they could ask to draw and label the structures. Okay, the next part of the question is, I think I have missed maybe some information required for this question. celiac disease result in part C containing fever villi. There is a disease called celiac disease result in part C containing, they say the villi number decrease, the celiac disease. So probably I have to check this one, whether this particular question, the purposely they did not give information, because I remember when they write the diseases which you don't know, celiac disease, they give information. Now the celiac disease, there is certain food components. For example, there is a food called gluten. So this gluten, people have allergy for gluten. When we eat gluten, then our protective system try to attack gluten molecule, which cause a problem in the villus. Then our protective system, they try to destroy villi. Then the villi number decrease. That disease is called celiac disease. Suggests why celiac disease may affect the growth of a child. Only thing you can understand,
because
fever
when the
villi.
villi are, sir, because when the villi are decreasing, the surface area is decreasing, so there is less absorption of nutrients.
Yes, the main thing is we learn villi, they increase surface area, right? So we learn this villi make projection like this, which increase surface area. When surface area increase, they can absorb nutrient faster, and they also have a rich capillary and also lacteal. So lacteal, capillary, surface area. So they are the three main components, increase absorption. Then celiac disease, what it says, the person lost a large number of villi, so they may have like this, kind of this. So diseased person have less number of villi, right? So answer is yes, you can write in celiac, when there are fewer number of villi, the surface area decreases. When there are fewer number of villi, surface area decreases. When there are fewer number of villi, surface area decreases. Then there are also less capillaries. And less lacteal. Less lacteals. Less lacteals, that's this one. Lacteal absorb glycerol and fatty acid.
Sir, can you repeat that sentence?
Then less capillaries and also less lacteal. Less capillaries and less lacteals. Next, less nutrients absorb. Less nutrient absorbs. Less nutrient absorb. Then the growth decrease. Then the growth decrease due to lack of nutrient and energy. Due to lack of nutrient and energy. Okay, next week homework, you can do question number 5. Question number 5 for next week. Right, the diseases, so there are 2 type of diseases. One is called infectious, other one is called non-infectious. So we can divide all the diseases into 2, infectious and non-infectious. Now infectious diseases are caused by pathogen. So the meaning is, if someone is infected and this person can transmit the pathogen to another person, then this person get infected. But other diseases, this person is have the disease, but the person cannot transmit the disease. Here can. So infectious disease caused by pathogen, non-infectious disease caused by other factors. They are not in pathogens. For example, the heart attack, cancer, diabetes, or maybe some kind of a psychological disorders, the depression, autism. So these kind of things, they cannot transmit to the others. But these diseases, they can transmit. Now, I think you are the good victims of infectious disease during COVID-19 time, because you have seen things very nicely. And when the infectious disease, this kind of a people, if they can spread the disease, we have one method to control it. These people we're going to isolate. That's called quarantine. So we call it quarantine. The word used, quarantine, to isolate the persons. For example, COVID-19 time, there are centers, and they take these people to these centers, and they have to stay until they get cured, or 7 days or 10 days like that. They get the treatment. They try to take out from the society, and they have to stay there. Nowadays, we also have this. For example, simple thing is, if you have a flu, so better you stay at home. If you go to school, you can spread the disease. So if somebody has a flu, work at home, stay at home, not going for work, because they can infect the disease. So we have this kind of a standard. If the disease is quite dangerous, then they're going to quarantine these people quite strongly. So COVID-19 is one thing, and there is another disease. This disease is called Ebola. So Ebola is also caused by virus, and it's a really dangerous disease. This outbreak came 2012 in Nigeria. There also they have to quarantine people to spread the disease. Now, this I have a kind of a special situation. there is a person, this person had lifetime quarantine. Lifetime mean like it's just like a jail, lifetime jail. They keep this person lifetime. So why this has happened? The person quarantine lifetime? Some diseases, people don't get the disease. They look healthy. They are healthy, but they can spread the disease to the others. So there are some infectious disease. The person never get it, but they can infect. That means bacteria living inside the person, this bacteria cannot make the disease of that person. So this, I will tell you soon what is the disease and how this has happened. And last thing, we're going to do a practical, right? There's a practical. Practical is use of podometer to measure transpiration. So don't worry too much when you have practicals, because we have too much practicals collectively, chemistry, physics and biology. But all these practicals, they never ask to write the practical. The question come from the main practical. They ask here and there questions. They don't ask to write completely whole practical. So they maybe worst case they can give part of the practical, and they ask what is this, why this use, why this use, why this use, like that. They're going to ask a question. Otherwise, if you add all the practicals in chemistry, physics and biology, it could be around 60. That's quite a lot. So, but because of that, they never asked to write the detail about the practical. But we have to understand the kind of important thing about the practical. But some practical quite okay. When you look at it, we can understand. Now I draw this podometer, how it looks like. So it looks like this. So these are glass tube, and there is another little bit more tube is going up, and here we have the tap or valve. So then this two end, we have a rubber stopper called bung. Right? So this is the rubber stopper, we call it bung, and this one is also called bung. Right? So that's everything is a glass then, the glass tube, right? That's a glass tube, and this is also glass tube with a small tap, right? Now what we do, we can fill a whole glass tube with water. Okay. So I am going to fill, don't have this red, okay, so I'm going to fill this with water. Right. Now to this end, I can also make the hole in this rubber bung, and I can insert another tube, the small tube, to this one, right? So usually water is not leaking. So when we have an idea that when we insert this to the rubber stopper, the rubber stopper is a very strong, it can again, because it's elastic, it can again push it, and usually water is not leaking. So if we fear about water is leaking due to small space here, we can apply some wax like things that prevent, because of any pressure comes, if they go, they can apply some wax like things around here. And this one is connected now to again water container, and here I have a ruler, and here is a bubble. So usually we can take a syringe with the air and then we can push it, then the bubble can go here and bubble can stay somewhere here, right? So now to this end, I again make a hole, and I am going to insert a plant, right. So now I set up everything like this, then I can check any factors. Let's say I place a light here. So when I place a light intensity is high, then usually the water is going up only here. Transpiration take place, then the water is going here. When water is going here, the bubble is moving. So the water is, water can come from this beaker, so the bubble is moving toward this end. So then I need a stopwatch to measure the time, right? So now let's say the bubble moves slowly to this end. Then what I do, I open this tap. When I open the tap, water is going this direction and the bubble can again move to this place. So I can repeat the experiment. So then again bubble is moving this direction. Once it come to here, then I slowly open the tap and then the bubble is going back to the same place, which is important for me to repeat the experiment. Let's say the bubble, that's a bubble starting here, and I start the stopwatch. Then after 5 minutes, the bubble come to this place. It took 5 minutes. Then I increase the light intensity. Then it take 3 minutes. So this method is useful to measure the transpiration by measuring the time taken the bubble move to the one end. So we will write something and we will see how it work in a kind of animation. Can you copy this down? So I will label these things. This is called capillary tube. That's called reservoir. Reservoir. The plant. That's a ruler.
Recording in progress.
Okay, shall we write how we can do this practical? Okay,
Sir,
number
I'm
one.
still drawing.
Okay. These people told online or three girls used to come. Why I asked them to come next time physically. No, they also
So they come back online.
they join online.
From now
Well, not
sometimes.
sometimes. I will ask next time to come physically. Others are from different places now. All from Ratnapura, Gampaha, Nigambo, Anuradhapura, all these areas.
Okay, sir.
This class only you are the one. I don't know where other student I couldn't find them. How do you get this contact?
I don't know.
Maybe your mother.
On Facebook.
What's that? Because I had some students like probably you know them Sasen.
Yeah, every day.
I thought that they have contact with the parents know each other. Sasen, Yash. There are a few like last year. Maybe they are going specific places. Okay, shall we write? So we can start writing number 1. Cut the plant. Cut the plant. Cut the plant diagonally. Diagonally is a slope or slant, not the vertical. Diagonally. That's double L. Should be double L, right? Diagonally. Underwater. Cut the plant diagonally underwater. So sometimes, you know, it's important to know why we cut diagonally. You know, that's a kind of a practical thing. If I want to plant a stem, or you know, I can cut a small stem from the plant and I can plant in another place. That's called asexual reproduction, mostly common in plant. We cut it like this, the stem. This increased surface area, then you know when we plant under the soil, they have more surface area to absorb water. So rather cutting like this has more surface area diagonally, that increased surface area. And why we cut under the water? So if you cut it, take it out and we cut it outside, there is a chance air can enter into the xylem. You know, air bubbles can block the xylem. Air bubble can block the xylem. So, for example, sometimes if we want to kill animals for various reasons, like small animals, for example, let's take a chicken. So if we want to kill a chicken in a farm to check whether chicken has infectious disease, what usual method is they inject just air to the blood vessel. So when we inject air to the blood vessel, immediately chicken die, because it can go to the heart and or blood vessels in the heart, and the bubble can block the vessels, blood vessels, and the blood cannot pump. So air actually stop in the middle. That the strong air bubble, then the liquid cannot pass. So same thing can happen if we cut outside, the air can enter and it can block the xylem. So then our experiment will not work. Then the second thing, then set up the photometer as shown in the diagram. Set up the photometer as shown in the diagram. Third one, use petroleum wax
Set the photometer.
as shown in the diagram.
Sir,
Yes? Yes?
sir,
Yes?
can you repeat the second one?
Set up the photometer. Set up the photometer as shown in the diagram.
Thank you.
Third one, use petroleum wax petroleum wax to seal any leak. And the fourth one, then measure the distance traveled by the bubble. Measure the distance traveled by the bubble with the time. Traveled by the bubble with the time. Okay, so that's end of this plant transport. And if you look at this tube, I have given you a practical. This practical. So, if you read, this was a past paper question. The question says, describe how to set up and use the student apparatus to test this prediction. Transpiration is affected by several environmental factors. Student investigate the effect of light intensity on water loss from plant shoot. The diagram shows some of the apparatus the student uses. Student predict that water loss from the plant shoot will increase as light intensity increases. And the marking scheme points are given. So it's a 4 marks question. So you have to actually write this practical. That's why I gave. So some practicals are important. If you see this kind of a thing, focus that there is a chance this kind of a question can come again, that you have to write. Number 1, cut shoot underwater, cut shoot diagonally, dry leaves, check seals, make sure seal, use petroleum jelly, bung is in, measure distance, bubble move, and in set time, started time. So then the other one is they use the lamp. So lamp is for us is not important. The particular question use lamp. Place lamp different distance from the shoot and started control variable temperature, humidity and wind speed. So today we learn light intensity, temperature, humidity and wind speed are the 4 factors affecting transpiration, and repeat the experiment. So these are the 7 points given to get a 4 marks for the question. So that's end of this session, and now I will show you a video about this experiment again, how this experiment is used practically, and then you will
Sir,
better idea. Yes?
why do we dry the leaves?
Okay, so good question, right, when you ask it. So let's say, so we cut this on the plant under the water. So when we cut under the water, this leaf is wet now. When the leaf is wet, then you know a lot of water. So that transpiration is loss of water. Then this water also move to the atmosphere. That's not real water. They are on the surface. So that's not good, because it bring error to the practical. The water which actually on the surface is not coming from the plant. They are coming from the water container that I use to cut the leaf. For example, I can cut the plant. This is the plant. So then it's completely filled with water. Then I cut from here like this. Then it's damp. Everything is, the whole plant is damp now. Then I have to dry it to remove the excess water. Otherwise, it might bring some kind of experimental error to the practical. Okay. We can see the screen. Let's see whether I can manage. Don't see applications. Oh, they have to share, you have to see, right? Okay. Cannot minimize. Okay. Ooh. Share. Okay, can someone confirm that you can see the screen, this not the whiteboard?
Sir, the screen is visible.
Right. You. It's not coming to the screen. Let me check. Wait a second. I think it's this. Yeah. No, it's okay. Yeah. Yes. Right, you can see how this animation shows how the things can go through xylem and phloem, and this is xylem and phloem, and we call them vascular bundle. And now this picture, I think it's better. So here you can see xylem and the phloem together. Now phloem, last time we learned, actually there is a plate here that's called sieve tube, sieve plate like things, and xylem, the blue color one, usually they don't have such a things in the middle. They make a continuous column, and they have a kind of a filter like things in the middle. Okay, this is the experiment, right? Photometer, we're going to learn that one. Right now, this person is filling the water to the beaker, which is attached to the capillary tube. And now he's cutting it under the water. So diagonally. So he's applying the petroleum wax or gel to prevent any leakage or loss. Okay, now I think he could insert the bubble. That's for the wind. Now you see the bubble is moving through capillary tube. Final reading and initial reading is the distance traveled by the bubble. Okay, that's the end. Now next thing is I'm going to share today's video. It's about a life lesson video that we last week also learned one. That's one we learned last week. Okay, it's not the one. Okay, that's the one with the. It's not this today's one is not the real one. It is an act, and you have to listen with the voice. I will also give the voice. Yes, qualities of good student. So we will learn one by one. Today's one is empathy and social skill. Empathy means how you feel someone emotion like him or her. That's very difficult. That's very really a difficult thing to actually get it. So some people they don't show sometimes. That's empathy. Whether they are sad or they are how their life is things, the situation, how the sad to him or her. You have to look as the person how we feel it. That's empathy. And social skill is you have to have so much of thing understanding the society, people, and communication. So this today's one actually explain that one, right? Let's watch. So. I don't know how good you get the sound. I have to actually make some kind of a changes to make the sounds. Share. Okay, it's not still sharing. Wait a second.
Pertaining B.
Share computer sound. Okay. Share computer sound. Okay, done. Yes. Oh, okay. Let's watch.
Twelve, 13, 14, 14B. Here we go. Oh, it's not fitting.
Oh, excuse me, you're rubbing all up against me.
Oh, I am so sorry. I'm just having problems with my bag.
It's not rocket science.
Oh, here, let me help you with that.
Thank you so much. I don't know why they make it so complicated.
They're not complicated, you're just not smart enough.
Sir? That's not very nice.
What? I'm just being honest. I mean, look at him.
You know, you should never judge a book by its cover. There we go. All set.
Thank you so much.
My pleasure.
Excuse me. Do you mind if I get in?
Do you have a choice?
Hi, my name is Tom. It's nice to meet you. Nice book. How you like it?
I'm trying to read. What did you say now?
Well, I'm sorry. I was just asking about the book you were reading.
Okay. Well, it is called Advanced Physics, and it is by Thomas Watson, one of the most prestigious physics scholars in the entire world. And I would explain it to you, but I don't think you're smart enough to get it.
Well, actually, I like physics. You see.
You know what? I really don't want to be bothered. If you have a problem, call the flight attendant. Okay, I'm not here to babysit you.
Hello, sir. Would you like some water? Hello again. Would you like some water?
Sir, thank you.
You incompetent imbecile!
I am so sorry. That was actually all my fault.
Don't take the blame for him. I knew he was going to be in trouble the second I saw him walk in. This is ridiculous. Nobody should have to sit next to this. You know what? I demand a new seat immediately.
Okay. I'll go talk to the captain and see what we can do.
Yeah, you do that. I'm glad we have an understanding.
Oh my God. I'm so sorry. Can I
No. Clearly, you are not smart enough to do anything right.
I'm sorry.
Okay. I've spoken to the captain.
Great. Did he find me a new seat?
Even better. He's upgrading the ticket to first class.
That's what I'm talking about. And thank goodness I don't have to spend the entire flight sitting next to you.
I'll just grab the suitcase.
Oh, no, that's not my suitcase. You got the wrong one.
Oh, I'm sorry I wasn't more clear. The captain is actually upgrading his ticket to first class. Because we all agree he shouldn't have to sit next to someone like this.
Wait, that's ridiculous.
Excuse me, sir. How would you like to move to first class with us? We'd love to have you there.
Wow. I love that.
Great. The captain will be over shortly to personally escort you. I'll grab your bag.
Excuse me. And by the way, that book is even better than the next chapter.
What are you talking about? How would you know?
Because I am Thomas Watson. I wrote that book.
Wait, you wrote it? I don't understand. Like you don't look like somebody who would...
Well, I guess you should never judge a book by its cover.
Mr. Watson, we have your seat ready for you.
Great, I'm coming. Have a nice flight.
Okay, so I hope You learn something from the video, isn't it? It's an act, but still should not judge by the situation or people. And we have to analyze, and we have to a little bit calm down to make a decision. Right. Next week we have a quite advanced lesson, the human circulatory system. We're going to learn animal transport, human circulatory system, the heart, blood vessel, blood, and heart, blood vessel, and the blood cells and blood of the human circulatory system. So question, we have a question as a homework, don't forget. So do the question number 5, and we will meet again next week. And also remember, that will be the last class for this year. 31st we don't have a class, so only next week we have a class. All right, then until next week, you can little bit study and enjoy still your holidays, and see you again next week, right? So I'm going to stop the class. Bye, take care.
Bye, sir. Thank you, sir.
Bye, sir. Thanks, sir. Bye, sir.
Wait, go.